Mechanism of gastrulation. Hello, welcome students. Today we are discussing about the mechanism of gastrulation. In earlier videos we have studied that gastrulation is a process by which blastocyst is changed into a gastrula larva with three primary germ layers. It starts immediately after implantation and this cells of blastodermic vesicle visibly move in small masses to their final and predetermined positions. So students, these movements are interdependent and are also called morphogenetic movements. These movements of the blastomeres are called morphogenetic movements. These movements change a hollow spherical blastula into a complex gastrula with three primary germ layers named endoderm, mesoderm, and ectoderm. The developmental fate of cells of each of these primary germ layers is determined to develop specific organs and organ systems of the individual, and that fate is same in all the triploblastic animals. So when we discuss about the mechanism of gastrulation includes the formation of following structures. Number 1, formation of endoderm. The blastodermic vesicle enlarges and cells present on the lower surface of the embryonal knob detach by delamination from the embryonal knob. The detached cells become flat, divide, increase in number, and form the endoderm inside the trophoblast of the blastodermic vesicle. We see that embryo in this stage is a tubular and which encloses a hollow tube which is called primitive gut or archenteron, which is lined by endoderm. So we should understand that the archenteron is the hollow tube cavity which is lined by endoderm. The part of endoderm located under the embryonal knob is called embryonic endoderm, which later forms embryonic gut, while the remaining part of endoderm, along with trophoblast, forms the yolk sac. If we want to query about the fate of endoderm, we see that endoderm is predetermined to form midgut or archenteron. From pharynx to colon, middle ear, gastric and intestinal glands, tongue, urinary bladder, respiratory tract, liver, pancreas, thyroid, parathyroids, and anterior lobe of pituitary gland. Pituitary gland is known as master gland, which is composed of three parts: anterior, middle, and posterior. So here we see that anterior lobe of pituitary gland is composed of the endoderm, thymus gland, and primordial germ cells. Number 2, formation of embryonic disc. The blastocyst continues to grow due to absorption of more and more uterine milk. The embryonal knob stretches and cells of robber start breaking off and dispersing. So the cells of embryonal knob form a regular layer called embryonic disc, which becomes continuous with the trophoblast. Embryonic disc is differentiated into cephalic embryonic and caudal regions. Formation of embryonic mesoderm. The formation of mesoderm starts at the caudal region of the embryonic disc, where cells undergo rapid proliferation and form a localized thickening of the embryonic disc. The proliferated cells later get detached from the embryonic disc and form the mesodermal layer between ectoderm and endoderm. Fate of mesoderm. It is determined to form most of the muscles, connective tissues, dermis of skin, peritoneum, skeleton, which contains bones and cartilages, circulatory system, heart, blood vessels, blood lymphatic system, excretory system except urinary bladder, adrenal cortex, and most of the reproductive system. So, this is the fate of mesoderm. Now, formation of ectoderm. The remaining cells of blastodisc become columnar and form ectoderm. An ectoderm is predetermined to form the epidermis part of the skin. Epidermal glands, fore and hindgut, pigment cells, brain, spinal cord, sense organs like eyes, retina, conjunctiva, cornea, lens, internal ear, nasal chamber, middle and posterior lobes of pituitary gland, adrenal medulla, and pineal. Now, the formation of amniotic cavity. Amniotic cavity is a space which appears between the ectoderm and the trophoblast and is called amniotic cavity, which gets filled with a watery fluid called amniotic fluid. It provides protection to embryo from mechanical shocks by acting as a shock absorber and from dryness. It acts as a shock absorber and prevents the embryo from dryness. At the floor of the amniotic cavity, there lie ectodermal cells, while at the roof of it, there lie amniogenic cells derived from the trophoblast. So, in this way, we see that there is formation of three germ layers, and these germ layers are destined to form different structural organs of the future fetus. We can summarize it in following way. We can name the structures formed by ectoderm such as epidermis, epidermal glands, hair, conjunctiva, and lens. By mesoderm, this is muscles, skeleton, circulatory systems, which contain heart, blood vessels, blood lymphatic system, kidneys, ureters, gonads, and adrenal cortex. While by endoderm, bladder, lungs, liver, pancreas, thyroid, parathyroid, thymus, anterior pituitary, etc. So, thanks for watching.